Imagine you're pulling a loaf of bread from the oven and marvel at those golden bubbles, and raising a pint of craft beer, admiring its creamy head, and realize both owe their magic to tiny fungal workhorses, yeast, whose chemistry drives baking and brewing in beautifully similar yet distinct ways. What exactly is yeast? It's a single-celled fungus, Saccharomyces cerevisiae, that consumes sugars and, in the absence of oxygen, converts them into carbon dioxide and ethanol, plus dozens of flavorful byproducts. In baking, the CO2 puffs your dough, while in brewing, ethanol becomes the booze. How does bread rising differ from beer fermenting? In baking, you mix yeast with flour and a little sugar, then keep the dough warm. The yeast feasts on maltose from the flour, producing gas that inflates the gluten network until you bake it. Heat kills the yeast, locking in the airy structure. Example, a sourdough starter, nurtured for years, carries wild yeast strains that produce extra acids and esters, giving that tangy crust. In brewing, you mash grains to liberate fermentable sugars, then cool the wort and pitch yeast. Over days, it gobbles maltose, maltotriose, and glucose, crafting alcohol plus hundreds of aroma compounds, esters that smell fruity, or phenols that smell spicy. Before flocculating and leaving the brew clear, example, Belgian ales use S. cerevisiae strains that produce high levels of phenolic clove notes and banana-like esters, while German lagers use colder fermentations for a cleaner profile. Why do temperature and sugar type matter so much? Warmer bread dough, 30 to 35 dSc, drives yeast into rapid gas production, ideal for a quick rise. Cooler beer fermentation, 10 to 12 dSc for ales, slows yeast metabolism, controlling flavor development. And some sugars, like maltotriose, ferment more slowly, stretching out your brew's fermentation profile and mouthfeel. Can you taste yeast chemistry? Absolutely. Smoked breads harness yeast's Maillard reaction in the crust for complex aromas, and barrel-aged beers pick up lactic acid from leftover yeast, adding tartness. Even wild-fermented farmhouse beers rely on uncharted yeast species to deliver layers of funk. What can beginners experiment with? Try a no-knead bread that ferments overnight at room temperature for deep flavor, or a simple 5-gallon homebrew kit where you control pitch rate and fermentation temp to see how yeast strain choice transforms a base malt into a crisp Pilsner or a banana clove Hefeweizen. In both baking and brewing, yeast is your microscopic chef, turning starch into structure and sugar into spirit. So by playing with yeast strains, temperatures, and fermentation time, you unlock a chemical art that's as much science as culinary craft. Yeast are single-celled fungi that generate energy by converting sugars into carbon dioxide and ethanol through glycolysis, followed by fermentation when oxygen is limited. In baking, the CO2 inflates dough, creating the light, airy crumb characteristic of bread, while ethanol largely evaporates during baking. In brewing, controlled fermentation maximizes ethanol yield and contributes flavor compounds like esters and higher alcohols. Understanding yeast's dual capacity for aerobic respiration and anaerobic fermentation is key to both processes. Saccharomyces cerevisiae consumes maltose in wort, producing about 5 to 8% alcohol by volume, and releasing CO2 that creates beer's carbonation. Yeasts are like miniature biofactories that can either run engines with oxygen, burning fuel cleanly, or switch to backup generators without oxygen, producing exhaust. Baking leverages rapid CO2 production at moderate temperatures, 25 to 35 DEC, to expand dough, whereas brewing optimizes slower fermentation at cooler temperatures, 10 to 25 DEC, to balance alcohol content and flavor development. Baking requires a quick spike in carbon dioxide, so enriched doughs may ferment only an hour or two, while brewing spans days to weeks for complete sugar conversion. In baking, ethanol's transient presence is incidental, but in brewing, it is the primary desired product. Recognizing these divergent goals explains why yeast strains and conditions differ between kitchens and breweries. Pizza dough ferments for 90 minutes at 30 DEC, doubling in volume, whereas an ale fermentation occurs over 7 days at 20 DEC, reaching 5% alcohol. It's like using a car engine for a short city drive versus a long highway journey. Both use the same engine but require different tuning and fuel mixtures. Inside each yeast cell, glucose is split into two molecules of pyruvate via glycolysis, producing a net gain of 2 ATP and reducing NAD+ to NADH. Under anaerobic conditions, pyruvate is decarboxylated to acetaldehyde, which is then reduced by NADH to ethanol, regenerating NAD+ for continued glycolysis. Concurrently, each decarboxylation step releases CO2 into the surrounding medium. This tightly coupled cycle allows yeast to thrive in sugar-rich, oxygen-poor environments like dough interiors or fermenting wort. In a 100 g portion of fermenting dough, yeast cells convert roughly 5 g of glucose into 2.4 g of CO2 and 2.6 g of ethanol before baking halts the process. It's like a factory assembly line where workers split raw material glucose into fuel, ATP, exhaust, CO2, and byproducts ethanol, then quickly reset for the next batch. Yeast enzyme activity, particularly that of hexokinase, phosphofructokinase, and alcohol dehydrogenase, is regulated by substrate availability, pH, and temperature. High sugar concentrations trigger glucose repression pathways that optimize fermentation over respiration. Osmotic stress from high sugar dough or high alcohol wort induces protective heat shock proteins and trehalose accumulation. These regulatory networks ensure yeast remain metabolically robust under the fluctuating conditions of baking and brewing. Brewers often oxygenate wort prior to pitching yeast to promote healthy cell growth before the switch to anaerobic fermentation. It's like a smart thermostat that adjusts heating or cooling depending on current outdoor weather and energy reserves, maintaining comfort without waste. Fermentation rate and flavor profile are highly sensitive to temperature. Warmer conditions speed up metabolism but can produce undesirable fusel alcohols, while cooler temperatures slow fermentation and yield cleaner flavors. Optimal pH around 4.5 maintains enzyme activity and inhibits bacterial contaminants. In baking, slightly acidic dough enhances gluten structure and yeast performance, whereas in brewing, pH adjustments during mashing and post-boil clarify the wort and support yeast health. Precise control of these parameters is critical for consistent product quality. A lager fermented at 12 DEC produces crisp, clean notes, while an ale at 22 DEC delivers fruity esters characteristic of classic English beers. It's like brewing tea. Steeping time and water temperature dramatically alter both extraction rate and flavor nuances. Yeast growth and gas production follow logistic kinetics. A lag phase as cells acclimate, an exponential phase of rapid CO2 output, and a stationary phase when nutrients deplete or alcohol accumulates. In baking, dough volume increases slowly at first, then surges as yeast enter exponential metabolism. Overproofing, exceeding the peak, leads to weakened gluten networks and collapsed loaves. In brewing, hitting an optimal yeast cell density ensures complete sugar conversion, whereas overpitching can shorten fermentation too much, reducing flavor complexity. A sourdough starter may double in volume in 4 hours at 28 DGSC, but then plateau as acidity and ethanol inhibit further yeast activity. It's like a fireworks fuse. It burns slowly at first, then reaches a rapid flare-up before fizzing out. Timing is everything to catch the bright burst. In baking, gas retention within the dough's gluten matrix determines crumb structure. Small, even bubbles yield fine, textured bread, while large pockets create rustic loaves. Yeast dread flavor compounds, organic acids, aldehydes imbue bread with depth, especially in long-fermented doughs. In brewing, yeast strain selection and fermentation regime shape beer's aroma. Phenolic, spicy notes from Belgian yeasts versus clean, neutral profiles of lager strains. Mastery of yeast chemistry is thus central to artisanal bakery and craft brewing alike. A slow, cold-proof retardation of dough overnight develops complex lactic and acetic acid flavors, enhancing taste and shelf life. It's like fine-tuning a guitar. adjusting string tension, finger pressure, and playing style yields vastly different sonic textures. Maintaining pure yeast cultures free of wild or bacterial contaminants demands stringent sanitation and regular viability testing. Genetic drift over successive repitchings can alter fermentation performance and flavor profiles, necessitating periodic strain rejuvenation. Balancing microbial ecology in sourdough or mixed culture fermentations adds complexity but offers unique sensory signatures. Robust quality control protocols, cell counts, viability assays, and off-flavor screenings are essential to reproducible results. A brewery's house ale yeast may be repitched up to 10 times before lactic acid bacteria cause sour off-flavors, forcing a fresh inoculation. It's akin to maintaining a garden. You must weed out invasive species and periodically replant heirloom seeds to preserve cultivar integrity. Advances in CRISPR and metabolic engineering enable custom yeast strains that produce novel flavors, enhance aroma precursors, or tolerate higher alcohol levels. Synthetic biology approaches aim to introduce pathways for diacetyl reduction, lactic acid production for sour beers, or even gluten-degrading enzymes in bread dough. Engineered yeasts promise to shorten fermentation times while expanding the palette of sensory experiences. Ethical and regulatory considerations will guide the responsible adoption of these next-generation microbial workhorses. Researchers have created yeast strains that synthesize monoterpenes, citrus aromatics directly during beer fermentation, eliminating the need for hops. It's like upgrading a car's engine to run on renewable biofuels and emit tailored scents, customizing both performance and passenger experience.